In this research, we simulated the extracellular matrix, or ECM, which is a gel layer that is secreted by and surrounds most fungi and bacteria. The ECM is the gateway for all metabolites which pass from microorganisms into substrate materials, and it is also the gateway which regulates what passes into microorganisms, ranging from nutrients to therapeutic drugs. The adhesive nature of the ECM plays important roles in problematic biofilm formation and in bioprocessing applications. We simulated the interaction between ß-glucan, a major component of the fungal ECM, and oxalate, a small organic acid which is secreted by many fungi, as shown in this animation. Interestingly, we found that oxalate binds to ß-glucan non-covalently to form a hydrogel. The formation of ß-glucan hydrogels with oxalate has not previously been reported, and we were able to confirm the nature of the bonding using Raman spectroscopy and density functional theory simulations. We know from earlier research that fungi can control oxalate synthesis and secretion, and in our current research, we found that the amount of oxalate secreted directly impacted the rheometric properties of the hydrogel. As oxalate was introduced in increasing amounts, hydrogels of increasing viscosity were formed. Further, the hydrogels impacted how model compounds diffused through the synthetic ECM. Oxalate was bound tightly to the hydrogel, and we found that diffusion of oxalate out of the hydrogel into substrates was limited. However, a high molecular weight protein simulating an extracellular enzyme was unable to diffuse through the hydrogel as depicted in the animation. Our results have practical implications. First, in fungi, extracellular enzymes would be diffusion limited within the ECM hydrogel and be unable to reach substrates. In brown rot fungi, this suggests that non-enzymatic mechanisms, such as the chelator-mediated Fenton system shown in the animation for depolymerization of lignocellulose substrates, are important throughout the decay cycle rather than just in the initial stages of decay. The ECM hydrogel does not limit the diffusion of catecholate metabolites involved in the chelator-mediated Fenton reactions. Secondly, in both fungi and bacteria, the penetration of therapeutic agents intended to inhibit or kill pathogenic microorganisms has previously been reported to be poor because the ECM is a barrier to the diffusion of these agents. Controlling the biosynthesis of oxalate or other small acids by microorganisms could be important to promote diffusion of these compounds through the ECM to allow better passage of drugs into target pathogenic organisms.